Thank you.
You have your,
I think so. Can you hear me? Okay. Hi everyone, my name is Nikita Alexeev. I'm from George Washington University and I'm going to present our work about genome rearrangements and especially estimation of true evolutionary distance and rate of transpositions. So what genome rearrangements are? For example, this is a reversal. I don't know if you can see that it's flip, so there was a chromosome and a reversal is an event when flip a part of chromosome like that. Or another example would be translocation, then to chromosome exchange their like tails. Let me notice that both these events, they kind of break chromosomes in two places and then glue them in a new order. So this is basically the definition of genome rearrangements which we are working with. And the typical question would be something like that. There are two genomes. They have a lot of common genes or synteny blocks. And typical questions are what was the evolutionary scenario between them? How one was transformed into another? It's a pretty old picture from Pavel Pevner's paper. It's mouse and humans X chromosome. And you can see this colored pieces are assumed to be genomes, and these black segments are just adjacencies which are connecting them. And you can see on the first move was rotated this small part when it was rotated like this whole segment, etc., etc., and we transformed humans X chromosome into mouse X chromosome. Okay? And typical questions are like that, given two genomes, what's the minimal distance between them? So what's the minimal amount of such operations needed to transform one into another? And this question is actually, it's based on Parsimony assumption, which actually assume that everything which happened in the shortest possible way, right? And what we are trying to do here is to estimate the actual amount of operation which are needed. And I was going to illustrate it somehow, and I borrowed this. It's a Rubik's cube. If you're all familiar with it, right? So this is completely solved. I borrowed on reception. I was lucky and they had it. So it's completely solved Rubik's cube, and you could randomly rotate it, and you could ask questions like, oh, in how many moves I could solve it back, or you could ask questions how many moves I actually did. There are two different questions. I mean, if I did just one move, you need one move, and I did my one move. But if I did I don't know, 10 moves, it probably can be solvable in 7 steps, okay? So this is the difference between these two questions. And Rubik's Cube it's a small example. I mean, in our problem, it's exactly like that, but instead of 3 times 3 times 3 Rubik's Cube, we have something like 1,000 times 1,000 times 1,000, and it's even not 3-dimensional. You could think about something in 1,000 dimensional, there are 1,000 pieces. But except this, it's just more or less the same problem, okay? And well, in order to do that, we need to represent all our data somehow. So we use so-called breakpoint graph. This is supposed to be a genome. In all our studies is usually about 1,000 genes, but in order to show it on the slides, it's just a toy example. It's a genome with 4 genes. So this colored parts are genes, and these black parts just illustrate that they are adjacent to each other. So this was our genome, and this is how we represent it as a genome graph. I hope you can see the difference between this bold lines which represent genes and this gray lines which represent adjacencies, but the bold one, I will erase them soon, so if you don't see the difference, it doesn't matter so much, but it's like alternating right now. I don't know. Can you see that? Okay, great, thank you. So we have two genomes. You can see that like this part, this segment 2-3 was flipped. So this is my reversal step here. We represent both genomes like that, like I described before. Then we draw both of them on the same picture. I'm not sure if it was possible to trace it, but it's just both genomes, both genome graphs are drawn on the same picture. And when we erase, so this, again, with bold lines represent genes, and it turns out we actually don't need them, so we just erase them. And this is a breakpoint graph, the main tool we use, the main data structure we use in order to analyze all these evolutions driven by genome rearrangements. This graph is pretty simple. It's just each vertex has a degree 2, so it's just a collection of cycles. Okay? And sure.
Why are these cycles? Yeah, that applies at the ends of the chromosome.
Oh yeah, this is a good question. So let me go back to this part. It's just easier to work with cycles. So this was like a normal chromosome, and we add this. I don't know if you can see that. It's like artificial block which make it circularized. Thank you. Yeah, it's a technical detail, but it's just way easier to work with circular chromosome, like circular genomes. And actually, we show that like all the estimated parameters would be more or less the same. I mean, it doesn't bring a lot of noise into the model. Thank you. Yeah, for this question. So yeah, so now they all have degree 2, so it's a collection of cycles. And this, again, it's the main object we work with. And so what happens if some reversal occur? For example, here, so reversal is, again, it cuts the genome in two places and then glue them in a new order, so it's modeled by so-called double cut and join operation. You cut it in two places and then join them. So here, these two red edges were replaced with these two red edges. It's how one, say, reversal looks like on breakpoint graph. So it's basically what's going on with breakpoint graph, and some genome rearrangement appeared. Well, this is like what kind of statistics do we have? We know like the amount of cycles of each particular length, and the length here is amount of edges in the cycle divided by 2. It's because like they all have an even amount of edges, the same amount of each color. We know the total amount of cycles. We know total length of all the cycles. So I have mentioned that the minimal distance is like well studied, and just having this information, we can find the minimal distance by this formula provided by Yankopoulos and co-authors in 2005. So you just find the total length of all the cycles and subtract the amount of cycles. And there is a simple explanation under that, but I'm not going there. So, but the point is, just having this breakpoint graph, we can easily estimate the minimal distance between two genomes. Okay, and we want to use like the same statistics, but in order to find the true evolutionary distance, the actual amount of moves happened during evolution. So for two genomes P and Q, we assume that they have their common ancestor, and we model it. So we assume that there are like fragile parts in genome, actually the parts that can be broken by my rearrangements, and we consider the whole evolutionary process as a Markov process between genome P and genome Q. So it starts with genome P, and then it's with a series of DCJs of this transformation transformed to Q. Okay. So how it looks like in the very beginning is just a collection. So these two genomes are the same, and it's just a collection of cycles of length 2, of length 1, with our definition of length. Okay. we call them trivial cycles. And now you could imagine this random moves of Rubik's cube. So we try to perform our to model like our rearrangements like that. So first rearrangement broke these two edges and create this cycle, and something like that happen, and something like that happen, and something like that happen, something like that happen, etc. Okay, it's again it's a small example in our. We usually assume it's about we usually consider it's on about 1,000 pieces. Here were just like 5 of them. So that's basically what's going on. And in the end of the process, we just have this breakpoint graph, and all what we know is the length of all its cycles. And there are like two models applying to this situation. In one model, we say that it can be, the genome can be broken in any two places with equal probabilities. I mean, in any two places except genomes, except genes, because genes are under negative selection. In our model, yeah, give me a second. In our model, we just consider that there are like some fragile regions in the genome. So that's, I will explain how it reflects on the whole model. Yes, please, was the question.
Exactly. So when you said parsimony versus actual, so actual is the maximum likelihood, so it relies on some model, and this is your model, just uniform, random, uniform.
That's true. Yeah, yeah, and I will discuss a bit like pros and cons about that.
Yeah, but so you optimize the likelihood of this.
In a sense, under fragile breakage model. Sure. Yeah. But I assume that any two fragile regions can be broken, like a pair of fragile regions can be broken with like equal probability. It doesn't depend on the particular pair. But what's the difference on the level of my breakpoint graph? The difference is, so consider I get this, this is a breakpoint graph I get on my last like step. And you can see that this part were presented in both genomes, like red adjacencies correspond to genome Q, black adjacencies correspond to genome P, and you can see that they presented in both. And we say we don't know, is it just because it wasn't broken randomly, but it can be broken, or it's probably also under negative selection and it cannot be broken like at all. So we say that we don't, like, we have this breakpoint graph, and we don't know how many of these trivial cycles are there. We consider it like that. Okay, so this is the difference between random model and fragile model. It's, well, basically I'm just saying I don't know how many solid regions and fragile regions are there in my in these genomes. So, and it's reflected like that. Well, so in this model I have a set of observable parameters. Observable parameters are mostly the amount of cycles of each length which is equal to or greater than 2. Because cycles of length 1, I consider them hidden. And like derivative of these numbers. Total length of all the non-trivial cycles in the breakpoint graph, and the minimal distance obtained by the formula I showed you before. Well, and we don't know, again, we don't know the amount of trivial cycles, we don't know the amount of fragile and solid regions, and we don't know how many actual transformation occurred there. So this is this is our main goal to find, and this is just parameters which we cannot use. They're not observable in our model. Well, and here it's a mostly mathematical result. We have this nice formula. I actually believe it's nice, and I will explain why I think so in a second. So we obtain the result that the amount of cycles of a particular length, say L, times L over N, over the total amount of fragile regions, have this behavior. This is actually a really nice formula. It appears in a lot of, we didn't know that before we got it, but it appears in a lot of different mathematical models, including Erdős-Yi random graph, including like Poissonian branching processes, stuff like that. And the correspondence between our model and these models are, I mean, we can trace it, but it's not very obvious. Like it's all connected to each other. And with these formulas, we can also estimate like parameters. Let me also demonstrate why this formula is good. So consider all the possible cycles from length 1 to length, I don't know, 1,000 or like whatever. If you add all of them, it should be like the sum of these numbers should be 1 because it's like the total amount of edges over total amount of edges, right? It should be 1. This is not always true because it holds only for cycles of fixed length, so it's not hold for like infinite length of cycles of length compared to the size of the whole graph, and you can see it in this picture. So while the total amount of moves was pretty small, less than 1, all the cycles were small, and it was like constant equals 1. And then after this threshold, it goes down, and some giant cycles appear in my breakpoint graph. And also, it also helped with problem like minimal distance versus true evolutionary distance. So on this graph, you can see like it was on simulated data. So this was the actual amount of moves we performed, and it was like estimated by the formula the minimal amount of moves. So you can see that at least before 1, there is no big difference between like parsimony approach and like true evolutionary distance approach. They are just the same. But if you have like a genome with 1,000 fragile regions, and there were like, I know, 300 reversals or genome rearrangements, we are somewhere here, and like it's safe enough to say that we are in parsimony phase. But if it was performed, say, 1,000 moves, we are somewhere here. Oops, there's somewhere here, and it's about 15% different between like true evolutionary distance and minimal evolutionary distance. So we underestimate that about 15–20%. Yes, please. Oh, gamma, yeah. Gamma is just, Gamma is just the proportion of, so K is the amount of moves, N is the amount of fragile regions, so gamma is 2K over N. That's how okay, yeah, thank you. It's just like the relative amount of moves relative to the amount of fragile regions. Twice this ratio. Twice the ratio, yeah. Yeah. So it like, when like, if an average everyone was broken, it's not parsimony anymore. It's very rough statement, and it's like, formally it's not correct, but it's how you can think about that. Yeah, thank you. So this is like, if you perform the amount of moves is less than a half of total amount of fragile regions, you are in parsimony phase. If it's more than, you are not. And we don't know this n, right? It's an amount of fragile regions. So this picture, this graph doesn't help us to actually find gamma. And as soon as we find gamma, we can find everything else. I will show how. But this is a ratio of minimal distance over the total length of observable graph, the total amount of edges in observable graph. It's a monotonous function. So, and both of these parameters are observable. We know both of them. So as soon as we have two genomes, we can evaluate D, we can evaluate B, and we can like find the ratio, and then we can find gamma like that in a unique way because it's a monotonous function, right? And as soon as we find gamma, we can find like the amount of everything, of fragile regions and the actual amount of moves which were performed. And we run our our methods, first of all, on simulations. And it shows, I'm pretty sure now that you can see the numbers from there, but it shows that like the relative error is always, it's always less than 10%, and for like small gammas, so if you're close to Parsimony phase, it's less than 5%.
So what is the error here? Is the actual modifications you did?
I mean, it's on simulation. It's on simulation. So I do know the amount of.
So the difference between what you compute and
Yeah.
what you actually
Over what I actually, So it's a relative error. This is it. So it's what I estimate minus what I compute over minus what happened in like simulation over the amount of of moves happen in simulation.
and it converges.
It's
Can you show some consistency here?
Oh, it converges. I mean, If n goes, if the amount of fragile regions goes to infinity, it's convergent probability. We have like in the paper we have like all the bounds of like of the error, and we also performed it on 5 IST genomes, and we show that like for some of them. So in this table there are like 4 IST genomes, so this number is the estimated true evolutionary distance. This number is minimal distance. So, and we show for some of them they are in Parsimony phase, and for some of them they're like 5-10% over like Parsimony phase. So, in some cases we minimal distance underestimate, like Parsimony approach underestimates the distance by 5-10%. How much time do I have? Like 5 minutes, right? Is that correct? Okay. Like one of the restrictions in this model is that actually it's not very fair to assume that any 2 fragile regions can be involved in the same rearrangement. And like here, it's a schematic picture of DNA, and I don't know if you can see that, but the point is that if 2 DNA regions were close to each other, the rearrangement can happen. If they were far away from each other, it's not the case. So now we are working on like on this issue, and we take like high C data in order to estimate the distances between like DNA regions and trying to incorporate it into our model. This is one of the issues. Another question we can answer this in the same fashion is the question about transpositions. So transpositions are our like a different type of genome rearrangement. So it looks like what happened? There are like 2 chromosomes, and during this event, a part of one of them is inserted into another one. I don't know if I hope this picture is pretty self-explainable. And it requires free breakage of a chromosome, right? Like here, and there. And on the level of my breakpoint graph, the difference is that now I break not 2 red edges, but 3 of them. So it's kind of like changes the situation a bit. In particular, there were like there's a classical, not classical, but not very recent paper of Pevner, where he showed that the amount of moves, if you allow these transpositions, it allows you to like move faster between genomes, right? Because you can kind of do more, right? And this is the amount of moves you need. It's the total amount of fragile regions minus the amount of odd cycles, which means cycles with odd amount of odd length, over 2. So, like in the best or worst case, like how do you like better? You can move like twice faster than if only like double 2 break are allowed. But the problem is that these events they're known to be rare. So it's not fair to say, okay, we can just move twice faster because the proportion is like the proportion can be. It's important. Like people do not believe that all evolution are formed by this transpositions. They should be rare comparing to like reversals. So we can actually estimate using the same actual technique. We can estimate this proportion of transpositions. Again, we in the same circumstances. There are like a certain amount of fragile regions when we don't know this amount, but we treat it as a unknown parameter. There are a certain amount of two breaks, which means basically reversals on translocations. There are several amount of transpositions or free breaks. So these are my unknown parameters, and we are trying to estimate it with a given breakpoint graph, which is with a given amount of cycles of each length greater than 2. And again, we can estimate like everything, like the amount of cycles of length 2, the amount of cycles of length 3. In this case, it's just enough. The problem here, it's like, so we have like 3 unknown variables, we have 3 kind of known variables, we can solve this system of equations, but the problem is it's not a linear system of equations, so it's not always solvable. Oh, by the way, this estimations are pretty good. They fit, so you can see how well they fit like the simulated data. And solving this system of equation, we get some function like that. I don't want to go into detail, but you can imagine like you have you have 3 equations. You can solve them for 3 variables. Unfortunately, it's not a monotonic function anymore, so a solution can, first of all, it's not always exist and it's not always unique, but for the cases it's, for like a lot of cases we observed it wasn't unique and it existed. So this is like our like estimation for, so for a given, we perform simulation for a given rate of transposition and we estimate it with our method. And in most of the cases, the relative error is less than 5–10%. And we also do it for 3 mammalian genomes, and we show that in like what it was, rat and macaque, rat and human, and macaque and human. And in all these 3 pairs, the estimated probability of transposition were more or less the same. It was about a quarter. Well. Yeah, all this work was performed with my boss, Nikita Alexeev from George Washington University. And thank you for your attention. Yep.
You raised this issue that breakage is not uniform and it might have to do with the 3D structure of the chromosomes.
Mm-hmm.
Do you have enough data maybe to try to infer it by using the phylogenetic context, so you could, in principle, try to reconstruct?
Oh, I mean, if you have like a lot of species.
And then maybe see whether certain.
Yeah, we were thinking about it. Like, if you ever have like a lot of species, not just 2, and they can, yeah, I can see that. Yeah, we didn't try it yet. We were thinking about that, but we didn't try it. It's a good idea, definitely. Yeah, thank you. Yeah. Yep.
Is it possible to incorporate in the model somehow duplication?
It is possible, but not like in a straightforward way. So in this study, we assume like the gene contact was kind of unique, which means like there was 1-to-1 correspondence between genes in both genomes. Yeah.
So when you have the proportion between reversals and the translocation,
Mm-hmm.
you still can uniquely and deterministically correct the parsimony to actual?
Well, and like for this part, like the part that I estimate like the amount of transposition, I can do that until it's not too far from parsimony phase. So I can do that actually, but if it's like, if this parameter gamma is larger than 2, if I remember correctly, it's like going to, it's getting worse.
So gamma is the ratio between the 2 operations? It's not. No.
No, it's not the ratio between 2 operations. It's like the proportion of transpositions. So it's like the amount of transpositions, all the amount of transpositions plus amount of the reversals.
Okay.
Yeah. Yeah.
But it looked like in your human comparison, this proportion of transpositions worked.
You mean here?
Yeah. So are you trying to suggest that about a quarter of the rearrangements?
Yeah, I mean it looks like in this analysis it looks like that. Probably it's too much, right? Yeah, it's probably too much. But you know, like, again, it's first of all, it's probably because we have like this box plot is actually pretty large. So in the most interesting part of this chart, the relative error is about 10%. This is like the first thing, and the second thing is probably for transpositions we actually need to take into account this 3D proximity. Oh, I don't know how to do that. So if you know, and if you can teach me, it will be really cool. The amount of scenarios is like huge. It's called like combinatorial explosion. So it's like we couldn't find the actual scenario. We could find mostly like its length. Yeah, yeah, just a number, but not like we couldn't see like all of them because it's like a huge amount. You could first of all you could permute it. And then sometimes you can do something. So it's at least the amount of permutations, right? Yeah.
So Nikita, so one practical, actually, not so mathematical.
Okay.
Like today, in sequence evolution, in sequence distance, no one considers the Hamming distance, which is the parsimony. Okay?
Mm-hmm.
And everyone corrects Jukes-Cantor, HKY. Would you suggest, it's not objective, would you suggest really to move from parsimony to your measurement? And in genome arrangement?
Well, I would say that it looks like you mean for string like evolution, right? If you consider like SNPs, right? It looks like parsimony phase lasts much longer. For a reason, like I mean, it's the space of all possible mutations are kind of larger than the space of all possible rearrangements. But I mean, I'm not sure about the status of this statement. I'm not sure about like is it the case or not. But as far as I understand that, well, it's a good idea to try to do something non-parsimonious, but I don't know how to apply exactly this thing to there. Yeah, but I hope it's possible. Thank you.